Yeah, I'm Filip Scheperjons, and first of all, thank you very much, Martha, for your friendship and support over the years, and for this invitation. I'm working as a neurologist in Helsinki. I'm originally from Germany, but for 17 years in Finland. And I got to know Martha because we published the first study on microbiota in Parkinson's disease in 2014, and since then have done further studies in this respect. And so Martha asked me to give an overview on our current concept and knowledge of microbiota in Parkinson's, and how this has also led to first clinical applications. Does this work? Oh, this doesn't work. So.
No, you have to advance on that.
Okay, all right. That's my disclosures. So, since most of you are not neurologists, I thought maybe we get into the basics of Parkinson's just quickly. So on the left side is a picture of a, typical, if that is actually existing, Parkinson's patient. So Parkinson's is the second most common neurodegenerative disorder. Prevalence is about 1% in people over 65 years of age. And it's considered primarily originally a movement disorder because that's the situation when we diagnose it. So when the patients start getting the movements get slower, muscle stiffness, and to a certain degree, many times also tremor emerges. But the concept has changed over the recent years, obviously, and I think we can, with quite good confidence, say it is actually a systems disorder. So the whole body is affected, and according to the current concept, mainly because the autonomic nervous system that innervates all our inner organs also degenerates, which leads to dysfunction of many organ systems, and that manifests in a broad spectrum of what we call non-motor symptoms, and that's shown in the picture on the middle. So those encompass brain symptoms such as psychosis, depression, anxiety, for example, hallucinations, visual problems, reduced sense of smell, and then problems with blood pressure regulation, heart rate, reduction of heart rate, variability, and to a quite large extent gastrointestinal symptoms, and the most frequent being constipation. And so this has been now studied quite intensively over more than a decade, and but there is still, in many cases, there have been problems fitting all the patients into the picture of what we thought is the initial steps and the progression of Parkinson's disease. And there has been, in my opinion, a great and progress made by Per Bockheimer and his group a few years ago, and I think we should also take this into account in our discussions here, that Parkinson is very heterogeneous. So really, there is not two same Parkinson's patients, and to try to disentangle or to understand this heterogeneity better, Per Bockheimer and his group, they did a lot of experimental and also in vivo imaging work and also pathological work, and came up with an hypothesis that is quite well supported by current research, basically splitting the Parkinson's population into two subtypes. One being the body-first Parkinson's subtype, where the neurodegeneration starts most probably in the enteric nervous system, and then spreads upwards through the lower brainstem, which leads to REM sleep behavior disorder, which is a certain type of sleep disturbance that is very specific for Parkinson's disease, and then further spreading upwards, and when it reaches the midbrain, then basically the Parkinson's symptoms emerge. So basically, meaning that these patients have already a widely spread degeneration of the autonomic nervous system before actually the Parkinson's symptoms start. And those patients tend to have rather symmetric motor symptoms. They tend to have a higher degree of cognitive impairment, and they tend to progress faster, and they have what we call isolated REM sleep behavior disorder, so sleep disturbance before the onset of motor symptoms. On the other hand, there seems to be a subgroup that was named brain-first Parkinson's, where the pathology starts in one brain hemisphere, apparently in the limbic system, from where it then spreads mostly unilaterally, so on one side of the brain, downwards, reaching the midbrain quite early, so when leading to the dopamine reduction, and then spreads downwards. And at the end of the day, after many years, probably it looks more or less the same. But the difference is that the sequence of events is so that you get the motor symptoms earlier because it reaches the midbrain first, and they are more asymmetric because it starts in one brain hemisphere, and they don't have the isolated REM sleep behavior disorder before the onset of motor symptoms. So those are clinical cues to this bipartition. This seems to be a more benign type of Parkinson's. And this is a crucial thing to remember when we talk about Parkinson's, that it's not the same across different people, and there's really important pathological differences here. And so why are we interested in the gastrointestinal tract? At least why did I get interested in it? And that's basically shown in this graph. So this is the typical trajectory, if this is existing, of a Parkinson's patient or of Parkinson's patients, where at the time point 0, basically the motor symptoms start emerging, so something that we can see. And then the neurologist would usually make the diagnosis maybe 1 or 2 years later, and then the disease progresses, obviously getting more and more symptoms, partly due to the progression of the disease and partly also to a higher risk for side effects of certain medications. However, when we look at the left side, that is what happens before Parkinson's is being diagnosed and before there's motor symptoms. And I already talked about the hyposmia, the depression and sleep disorders, but on the far left you see constipation. And according to some epidemiological studies, constipation may precede Parkinson's disease diagnosis even by up to 20 years. Probably on average we're talking about something like 5 years. But idiopathic constipation is associated with a 2 to 4-fold risk of Parkinson's disease. So something is clearly happening in the gut in many patients, not in all, but in many patients, and that is why we're interested in whether that could be help us understand the earliest stages and the pathogenesis of Parkinson's better. So we have now behind us more than a decade of microbiota research in Parkinson's. And so we already have meta-analysis published, and I try to give you an overview on what has come out of these analyses. I won't go into all the individual studies. In the microbiota field, we were talking many times about diversity. And we can discern alpha diversity and beta diversity. So alpha diversity means basically the diversity within a sample, within a microbial population. So roughly you could say the more different types of bacteria you have in a sample, the higher the alpha diversity. And across many studies, we have the impression now that from meta-analysis that there's no constant signal of an abnormal alpha diversity in Parkinson's disease. So there's no difference to healthy controls in terms of alpha diversity. When we talk about beta diversity, that means the difference between two samples, the difference between the microbiota composition of two samples. And that is usually measured in distances. So you can sum up like vectors, the different bacterial concentrations, and then you calculate a distance. There are many different methods to do that. And the higher the distance, the larger the beta, the larger the difference in the microbiota, and then we are talking about a beta diversity difference. And that is something that has been positive in Parkinson's across the board. So there is a difference in composition between Parkinson's patients and control subjects. And when you go further and you analyze what bacterial taxa are driving these differences, then this is a short list from one meta-analysis. So there is obviously some differences in the details to a certain degree, but there is definitely, let's say, bacteria that have been reproducibly associated with a healthy or a Parkinson's status. And for example, the most best reproduced findings are a reduction of short-chain fatty acid producing bacteria and Prevotella in Parkinson's disease subjects. And here on the right side, you see bacteria that have been shown to have increased abundance, and such as, for example, Akkermansia, bifidobacteria, lactobacillus, and desulfo vibrio has also been discussed, Escherichia, etc. So a longer list. And so these you can basically reproduce on a meta-analysis level. So we have quite good impression into what bacteria are on average differentially abundant in Parkinson's as compared to control subjects. But I have to say that all of these studies that I will show you, except for the last two, they did not take into account any of the body first, brain first differences, because those had not been described before. So what we can say is that these changes increase over time. So we did a study, and also others, of de novo Parkinson's patients. We did a collaboration with Dutch researchers from Groningen. Basically, we did 2 cohorts of de novo patients, so no medications, just diagnosed, and compared the microbiome with healthy controls. And the interesting finding was that when we did that, there was not a single specific bacterial taxon that was changed in both cohorts. But when you look at the functional potential of these bacteria that were changed, then what we saw was, yes, already at baseline, we see a reduction of short-chain fatty acid producing bacteria already in de novo Parkinson's disease. But the changes are more subtle than what we see in a more advanced disease. And that you see also in these studies from Italy and Japan. So here you have, that is a multidimensional scaling. So each dot represents one Parkinson's patient, one microbiome, and this is projected into a 2-dimensional space. And the further apart the dots are, the more different the microbiomes are. And you see that there is quite a lot of overlap, but when you calculate the centroids for these distributions, then you can see that when you have the healthy controls, they're clearly different, or more different than the Parkinson's population. And then you see that the more advanced the disease is, so de novo, early stage, mid stage, advanced stage, it goes further away from the healthy control. So you have an aggravation of the microbiome differences as the disease progresses. And basically the same in the Japanese study. They had also some REM sleep behavior disorder patients here. So, and 1, 2, 3, 4, 5 are the so-called Hoehn and Yahr stages. So telling us how far advanced the Parkinson's disease is. And the further advanced the Parkinson's is, the more dissimilar the microbiota is to healthy controls. So microbiome changes in Parkinson's increase over time. Of course, also medications of Parkinson's patients increase over time. So is this related to Parkinson's medications? And this has been looked at in different studies. And overall, it seems that most of the Parkinson's medications, including levodopa, have a quite minor effect on the microbiome composition. There's just one exception, which is entacapone, which is a medication that is not used on its own to treat Parkinson's, but that we can use to enhance the effect of levodopa. It's the so-called COMT inhibitor. And patients that are using entacapone, they tend to have, for example, higher levels of bifidobacteria and lower levels of Faecalibacterium in their stool. So that seems to be actually a drug effect and not a class effect. So other drugs from the same class, like opicapone and tolcapone, they don't seem to have the same impact. So it seems to be an entacapone-specific effect. So that is actually quite relevant for these analyses, but other Parkinson's patients don't seem to have, Parkinson's drugs don't seem to have that significant effects. So I'm, as a neurologist, interested in whether the microbiome is related to disease progression, because then my logic says that, okay, maybe if we can influence the microbiome, we could influence Parkinson's disease progression, because that's what we eventually want to achieve. So we want to do disease modification. And there's not too many studies that have actually looked at the connection between microbiota composition and disease progression. This study from Italy basically showed that when you have low levels of short-chain fatty acid producing bacteria, such as Roseburia in this case, then the Parkinson's progresses faster. And the same You can also see the other way around for lactobacillus, which is increased in Parkinson's, and the more it is increased, it seems, the faster the disease progresses. So basically, the bugs that are associated with Parkinson's itself also seem to be associated with the disease progression. Similar observation from our study in Finland. We saw that subjects that had Parkinson's patients that had low levels of Prevotella at baseline progressed faster over a 2-year period as compared to subjects that had higher levels of Prevotella, which seemed to be more in the stable group. So stable patients had higher levels of Prevotella as compared to progressed patients. So there is a connection between the microbiota composition and disease progression, prospectively. How about connection to symptoms? And there we have obviously motor symptoms, non-motor symptoms. So we had a look in collaboration with colleagues from the US onto the connection between depressive symptoms and butyrate and butyrate levels. And it turns out that actually the lower, in the Helsinki cohort, the lower butyrate levels patients had in their stool, and the lower levels of short-chain fatty acid producing or butyrate producing bacteria they had in the stool, the stronger depressive symptoms they had. So there's an inverse correlation between short-chain fatty acid and butyrate concentrations in stool and depressive symptoms. In terms of gastrointestinal symptoms, we chose in the Helsinki cohort to check whether our patients fulfill criteria for irritable bowel syndrome. So basically meaning a long-lasting change of bowel habits that's accompanied with pain. And it turned out that those subjects that fulfilled these criteria had lower levels of Prevotella bacteria in the stool as compared to subjects that did not fulfill criteria for IBS. And then in meta-analysis, it was also looked into connections between motor symptoms, which means MDS-UPDRS Part 3. So this is a scaling method that we use. So motor symptoms are connected to abundances of certain bacteria and also cognition, MoCA as well, also on a meta-analysis level. So there is connections to motor and non-motor symptoms with Parkinson's. Okay, with microbiota, sorry. So obviously, well, we would like to understand what are the mechanisms. Why is that? What causes these connections? And one method of trying to understand these dynamics is to do metabolomics. So basically, analyzing either stool or blood metabolites in with a less or non-biased approach. So trying to screen all the metabolites you get in a sample that fulfill certain criteria, and try to link that to bacterial abundances and to, for example, symptoms of patients. But this has been done in a few studies, and I have to say that unfortunately, I don't think we have, in contrast to the bacterial composition, in terms of the metabolomics, we don't really yet have a really story that is evolving. So different studies find different metabolomic different changes in Parkinson's disease, and there can be many reasons for that, including the disease heterogeneity, methodological aspects, and so on. So I think we're not really there yet in terms of really understanding what the metabolomic impact is, except for the reduction of short-chain fatty acids, especially butyrate, that has been shown across multiple studies in Parkinson's disease. But with other metabolomic pathways, I think we're not really yet there. Another aspect that may play a role and that we were interested in is epigenetic changes. So DNA methylation, which is one mechanism by which DNA or gene expression is controlled. And butyrate turns out to be a quite potent epigenetic modifier, and that is why we looked at, together with colleagues from the Van Andel Institute, into epigenetic changes in relation to butyrate levels in Parkinson's subjects. And I don't go into all the details, but the main point is that we find clear links between epigenetic changes in Parkinson's relevant genes, and in particular we find alterations in white blood cells, in innate immune cells, monocytes and neutrophils, not really in lymphocytes. So which may point to the fact that butyrate or changes of levels of butyrate in Parkinson's may impact innate immune cell function, so may impact inflammatory responses. And so when you then look into how the patterns of methylation across the genome is correlated with what is related to butyrate, that is how that is correlated with the patterns that are known for different diseases, then this is a very interesting correlation that you find. So basically, let's start with Parkinson's disease. So what we see is that there is a significant correlation of the butyrate-dependent acetylation, DNA methylation sites, and the sites that are known to be epigenetically modified in Parkinson's disease. Well, that makes sense because we looked at Parkinson's patients. But the interesting finding is that this is very closely related to the patterns that have been described in ulcerative colitis and Crohn's disease, but much different, for example, from patterns that have been described for Alzheimer's disease. So basically, this may point, this needs obviously further studies, but this may point to one mechanism that could connect Crohn's and ulcerative colitis with Parkinson's disease, because we know that these diseases lead to an increased risk of Parkinson's. And so the epigenetic modification or changes in inflammatory cells related to butyrate levels could be one potential pathway that is involved here. So, and this is like a summary of the current concept of how the microbiome may play a role in Parkinson's. It's a concept, so we don't know if that is true. So basically meaning that in a dysbiotic microbiota in Parkinson's disease, we see increased levels of pro-inflammatory bacteria, reduced levels of short-chain fatty acid producing bacteria, which leads to higher exposure to pro-inflammatory lipopolysaccharide, for example, and also to potentially aggregation-prone curly protein, which are bacterial proteins that can lump together and seem to also be able to induce clumping in, for example, alpha-synuclein, so making these amyloid clumps. So production of these, that altogether may lead to, seems to lead to a low-grade inflammation in the gut wall, and somehow, maybe related to the inflammation and to the curly, this ends up increasing levels of alpha-synuclein and pathologically conformed alpha-synuclein that then can induce seeding activity and basically causing the alpha-synuclein pathology to get initiated in the enteric nervous system and spread through the autonomic system to the brain, and also potentially inducing a low inflammatory state in the bloodstream, which can also impair blood-brain barrier permeability. Okay, since I'm running short on time, I would just like to tell you, I cannot go into all the details, but that we have now 2 studies that have looked into the differences of the microbiome between body first and brain first. So those came out last year. And at the end of the day, it seems that the changes that have been shown for Parkinson's disease in terms of the microbiome, as I mentioned, increased, for example, Escherichia, Akkermansia, and decreased short-chain fatty acid producers, decreased Prevotella, etc. That you can see the same changes when you compare body first and brain first. And the brain first seems to be quite close to healthy controls in terms of the microbiota composition. So basically, the differences that have been described for Parkinson's disease microbiota seem to be mainly driven by the subjects that belong to the body first subtype, according to these newer studies. So how have these things been so far translated to preclinical and clinical research? Well, there's a lot of preclinical data published. A lot of interventions have been tried. let's say, using more traditional probiotics such as lactobacillus, using butyrate directly, using prebiotics to feed good bacteria, etc. Obviously, with these preclinical studies, there's always the problem that, first of all, there's publication bias, so not negative results. It's more difficult to publish negative results than positive results. And we don't have good animal models for Parkinson's, so we have to be honest about that. So nothing has so far, unfortunately, we can stop Parkinson's disease in animals, so we're very good at doing that. I mean, I'm not myself, but humans can do that, but nothing has translated successfully to humans. So we are having a problem with our animal models. So that's why I'm always cautious about these. So I'm more interested in clinical trials and studies in humans. And obviously we're not that far, not so many things have been tested, but when we look, for example, at probiotics, there is quite a few probiotics interventions. So using more traditional probiotics, so which is mainly bifidobacteria, lactobacillus. And at the end of the day, what these showed that there is a moderate benefit of probiotic treatment on constipation in Parkinson's disease and quality of life, but not on motor symptoms of the disease. So that's like the take-home message for the probiotic studies. And then when what we were interested in is what we tried, and others too, is fecal microbiota transplantation. So basically, what you do is if you have a patient that has an abnormal microbiome and you think that the microbiome contributes to the disease, then what you want to do is you want to replace that abnormal bad microbiota with a good microbiota from a healthy person, and then you hope that this will take care of the problem, that the patient will benefit from it. And there's many different ways you can do it. You can give it from above, you could give it from below, you can give antibiotics before, you can give 1 dose, you can give 4 doses, 5 doses, whatever. So that is not really standardized. But it is one method that is considered safe and is relatively easy to do. And so that is why we have now seen, so to say, the first wave of these studies in Parkinson's disease. And in my opinion, there's only 2 studies that really give us evidence because they are double-blind, placebo-controlled studies. One is from Belgium and one is from Helsinki. So this is the Belgian study. They ended up having 22 patients in the FMT group and 24 patients in the control group. Control in this case was auto-transplant, or the patient getting their own microbiota back, basically. And this was given from above. And take-home message here, basically it seemed to be a safe intervention. And at 12 months, this Belgian study reported a relatively small but clinically significant improvement in motor symptoms in these patients. In Helsinki, we had 30 patients, 15 control patients. Placebo was just a single injection of glycerol and saline, and we did it through colonoscopy. And our patients were selected by a dysbiosis test in addition to other symptoms. So we looked at the microbiota composition and only took patients that had a Parkinson's type microbiome, so to say. And also a safe intervention in our case. Interestingly, also placebo, as FMT as well, changed the microbiome for up to 12 months. So the difference was stable. And interestingly, the dysbiosis state in the FMT group, after 1 month, 24% resolved to non-dysbiosis, but that reduced to 3% at 12 months. So basically, they went back to baseline in terms of the microbiota. Whereas in the placebo group, which is the most interesting finding here, the dysbiosis resolution actually improved over time to up to 33% at 12 months. So actually, the dysbiosis resolution worked better after the placebo treatment that patients got. And so that is reflected in our results. So we didn't see a benefit of fecal transplant as compared to placebo, but we actually saw an improvement of gut-related symptoms, so for example, constipation, in the placebo group, and also some mild benefits of motor symptoms at 6 months in the placebo group. And our hypothesis is what we're interested in is that the bowel cleansing that the patients went through before the colonoscopy, that this may have an impact on their symptoms. So I'm running out of time, so I would just leave the summary slide there. You can read it for yourself.